Hello, vetmates! Today I will be talking about the lactation in animals. Moms produce nutrient-rich milk to nourish their newborns, keeping them strong and growing. It's love delivered fresh. Lactation and the development of the mammary glands play crucial roles in ensuring neonatal mammals receive adequate nutrition and immune protection. Lactation enables the mother to nourish her offspring by producing milk, a vital process that has been optimized in both wild and domesticated species for survival and human use. The intricate biological mechanisms behind milk production involve the coordinated development of mammary glands and complex hormonal regulation. So, lactation provides neonatal mammals with nourishment without requiring them to seek food. The mother converts nutrients into milk, enabling her offspring to feed through suckling. Many domesticated animals, particularly dairy cows, produce more milk than their offspring need, allowing for human consumption and supporting a multi-billion-dollar dairy industry. Next, we have the mammary glands, specialized sweat glands. Mammary glands develop from structures called mammary ridges, which appear along the ventral surface of the developing embryo. These ridges extend from the armpits to the inguinal region, and the number of glands that form along them varies among species. In species like humans and elephants, paired mammary glands develop in the thoracic region, while in animals like cows and goats, the glands originate from the inguinal region. By the time of birth, the mammary gland consists of a system of ducts prepared to secrete milk through the teat or nipple. Now, I will discuss the diversity in anatomical position, number, and teat morphology among mammals. The anatomical structure and positioning of mammary glands, as well as the morphology of teats, exhibit considerable diversity among mammalian species. This diversity is primarily influenced by the evolutionary adaptations related to the species' reproductive strategies and environmental niches. In many mammals, teats are located in the inguinal position, a trait that provides effective access for neonates during nursing. The number of canals and ducts per teat varies significantly among species, reflecting their different needs and evolutionary paths. For cows and camels, both species have teats with two canals. In cows, each teat has one cistern that leads to two distinct canals, allowing for efficient milk delivery during suckling. Camels share a similar teat structure, supporting their unique feeding requirements. For ewes and goats, they exhibit a slightly different arrangement, with each teat having one canal that leads to a cistern. This single canal system is advantageous for their smaller offspring, allowing them to access milk effectively. For mares and sows, the mare and sow possess teats with two ducts per teat. This anatomical configuration allows for a more efficient transfer of milk during nursing, accommodating the larger litters seen in this species. In rats, mice, rabbits, bitches, and queens, in these smaller mammals, teats contain a more complex structure with 5 to 6 ducts per teat. This design facilitates feeding for multiple offspring, ensuring that each neonate can receive adequate nutrition. Lastly, for the primates and elephants, at the upper end of the spectrum, primates and elephants possess a significantly higher number of ducts, with 8 to 10 ducts per nipple or teat. This extensive duct system is particularly suited to support the larger size of their young and the increased milk production required for their developmental needs. Now we have the endocrine regulation of mammary gland growth. Hormonal changes drive the growth of the mammary glands through a female's life. Between birth and puberty, the mammary glands grow at the same rate as the rest of the body. However, after puberty, the glands grow more rapidly, stimulated by hormones like estradiol and progesterone. During pregnancy, additional hormones, including prolactin, play a key role in developing the alveoli that are necessary for milk secretion. Now we will know how the mammary glands grow significantly between puberty and pregnancy. Once puberty begins, the mammary glands experience a phase of rapid growth referred to as allometric growth. During this time, the ducts and alveolar structures, which are essential for future milk production, begin to form and expand. Estradiol plays a significant role in the initial development of the ducts, while progesterone contributes to alveoli formation during the luteal phase. Hormones like prolactin and growth hormone also accelerate the development of the mammary gland during this period. However, complete formation of the mammary gland structure is only achieved with additional hormonal support during pregnancy, preparing the gland for full lactation capability. Next, we have the final mammary development during pregnancy. The last stages of mammary development occur during pregnancy, particularly in the third trimester. Alveoli develop into lobules which collectively form lobes that produce milk. Hormones such as prolactin and placental lactogen prepare the mammary gland for lactation. By the end of the pregnancy, the mammary tissue is largely composed of alveoli ready for milk production after birth. Now we will learn about the lactation's role in immune protection and nutrition. Colostrum contains high concentrations of immunoglobulins or antibodies which are absorbed intact through the neonate's gut, offering passive immunity. This is especially important for animals like ruminants where the placenta is epitheliochorial, preventing the transfer of maternal antibodies during pregnancy. As a result, consuming colostrum soon after birth is essential for neonates like calves and lambs, who rely entirely on their first milk for their early immune defense. In contrast, animals like humans with a hemocorial placenta can transfer maternal antibodies in utero, allowing newborns to be born with some passive immunity. However, colostrum remains critical for neonates as the antibodies in breast milk further support the immune system, especially in primates, to fight off infections. How does the colostrum and milk contribute to the animal growth and survival? Well, breastfeeding has been linked to numerous health benefits for infants. Breastfed babies experience fewer ear, respiratory, and gastrointestinal infections compared to formula-fed babies. Furthermore, early breastfeeding has been associated with long-term advantages such as low risk of obesity, cardiac diseases, and type 1 diabetes in adulthood. For mothers, breastfeeding is linked to a reduced risk of breast cancer. In livestock, particularly calves, the intake of colostrum and milk is crucial for optimal growth, health, and long-term productivity. Calves fed milk rather than milk replacers show faster growth rates, lower morbidity, reduced mortality, and increased milk production over their lifetimes. The importance of colostrum, however, is short-lived, lasting only 2 to 3 days after birth. After this period, the milk's composition stabilizes with its synthesis increasing rapidly, peaking shortly after parturition and gradually decreasing. The dam's milk production directly influences the neonate's growth with conditions such as mastitis or agalaxia. Agalaxia is the failure to produce milk, potentially leading to neonatal growth failure. In this figure, we have the changes in the mammary gland as a function of reproductive stage. This will be discussed in the following slides. During involution, we have now the return to a non-secreting state. As the neonate transitions to other sources of nutrition, the mammary glands undergo involution, a process where milk production ceases and the glands return to their non-secreting state. This happens when milk is not regularly removed. This causes the pressure to build up with the mammary tissue, leading to the atrophy of milk-secreting cells. Immune cells help in tissue recovery, allowing the mammary glands to prepare for future lactation. Now we will discuss the hormonal presence and growth factors in milk. Milk not only provides essential nutrients and immunological protection to neonates, but can also carry a range of substances found in the blood of the lactating female. These include hormones, growth factors, and even exogenous materials like alcohol, drugs, and antibiotics, which can pass into the milk if consumed by the dam. This ability of milk to transport substances from the bloodstream has important implications for both animal and human health. Historically, this was evident when cows grazing on wild onions would produce milk with an onion-like flavor in the spring. The lipid-soluble compound from the onions would pass directly into the milk, altering its taste. In addition to flavor-altering chemicals, protein hormones such as prolactin, growth hormone or somatotropin, GnRH, and thyroid hormone or thyroxine have been identified in milk. However, these protein hormones do not have significant physiological effects on the neonate or human consumers, because they are broken down into amino acids in the gastrointestinal tract, losing their biological activity in the process. Did you know that there are steroid hormones in milk? Steroid hormones, including estrogen and progesterone, are also present in milk and are reflective of the female's ovarian cycle. Progesterone, in particular, can be measured in milk to determine the reproductive status of the animal. This has practical applications in dairy management, where cowside enzyme link immunosuppressant assay or ELISA technology can measure progesterone levels to assess reproductive cycling and diagnose conditions like cystic ovarian disease. Next, we have the advances in reproductive monitoring. Emerging inline assay technology in milking parlors holds the potential to revolutionize reproductive management in dairy herds. This technology would allow for the real-time monitoring of progesterone levels during milking, offering daily insight to a cow's estrous cycle, pregnancy status, and any ovarian pathologies. The ability to track reproductive health continuously would significantly enhance herd fertility management, improving both productivity and animal welfare. Next, we have the bioactive peptides derived from milk proteins. Milk contains over 15 bioactive peptides that have significant physiological roles, such as regulating blood pressure, preventing blood clot formation, and boosting the immune system. One group of these peptides, known as opioid peptides like casomorphins, is derived from milk proteins and exhibits morphine-like effects. Casomorphins may slow gastrointestinal transit time, providing antidiarrheal benefits and influencing amino acid transport, as well as the production of insulin and somatostatin. These peptides are also believed to have pain-relieving or analgesic effects and can induce sleepiness in infants. Though much remains to be discovered about the full range of their physiological effects, these peptides hold potential for the therapeutic applications due to their pharmacological properties. Next, we have the pharmaceutical proteins in the milk of genetically engineered animals. Advances in genetic engineering have enabled the modification of mammary glands in animals to produce therapeutic substances beyond the nutritional components of milk. A notable example is the recombinant human antithrombin produced from the milk of transgenic goats, which has been approved by both the FDA and EMA for treating a rare blood clotting disorder. The amount of antithrombin produced by these goats is equivalent to that of 90,000 human blood donations per year. Another example is recombinant human C1 inhibitor derived from rabbit milk used in the EU to treat hereditary angioedema. Additionally, other pharmaceutical proteins including fibrinogen, malaria antigens, albumin, and clotting factors are currently undergoing clinical trials by the FDA. The use of transgenic animals' milk for producing pharmaceutical proteins is a promising avenue for medical treatments and health interventions. Now we will talk about milk ejection. It is the moving of milk from alveoli to ducts for neonatal access. Milk ejection is the process that actively transfers milk from the mammary alveoli and fine ducts into larger ducts, cisterns, and eventually into the teat or nipple, enabling it to be accessed by a suckling neonate. This should not be confused with milk secretion, which refers to the synthesis and release of milk by alveolar cells into the lumina of the alveoli. Prior to suckling or milking, most of the milk is stored in these alveolar lumina and smaller ducts, where it remains due to the resistance of the fine duct network. Around 70% to 80% of the milk is held in these areas between milkings, necessitating an active ejection mechanism to make the milk available. Milk ejection is also a neuroendocrine reflex that involves sensory signals from the teat or nipple, the release of oxytocin from the brain or neurohypophysis, and the contraction of myoepithelial cells surrounding the alveoli and smaller ducts. These contractions push the milk into the larger ducts and cisterns for removal. Myoepithelial cells resemble smooth muscle cells and form a mesh-like structure around the alveoli, facilitating this process, known as milk letdown. This reflex is crucial for feeding the neonate and for milk collection in dairy operations, as it prevents pressure buildups and can lead to a reduction in milk production. That's it for now, vetmates. I hope you learned something from this video.